I wanted to tell everybody welcome to our webinar. And this is the first in a series of webinars in conjunction with the GRASP LGMD Consortium. So today's topic for the webinar is the status of LGMD clinical trials due to COVID-19. So the Speak Foundation has been asked by many patients, would we start doing webinars? Part of it was because they just had a lot of questions about the different resources that were available. And as you can imagine, patients were concerned and they had lots of questions about COVID-19. We have been inundated with these questions. Most of them have been about care, but the second most prevalent question has been about clinical trials and how the COVID-19 will affect clinical trials. So it seemed appropriate at this time that we would try to do a webinar. And our future plans is to offer more webinars in conjunction with GRASP LGMD throughout the year. So our future hopes is we've already got 2 more scheduled, and then we're talking to others about having others scheduled all throughout the year. So we're excited about that. And also coming up is Limb Girdle Day on the Hill. So that'll be LGMD Day on the Hill, and that'll happen later this year. And that is a total advocacy event that Speak Foundation is hosting. And Mr. Ralph Yanez, who's part of the Limb Girdle community, is going to be heading that up. And that's a really important advocacy piece for Limb Girdle Muscular Dystrophy. And just in case you didn't know, a lot of the limb girdle individuals have been in communication with the FDA about a patient listening session. And so that is in the works of being scheduled with the FDA. So patients will actually go in front of the FDA, and Speak Foundation is helping to put that together along with other organizations as well. And I want to thank Dr. Brad Williams for his help in putting that together. So I want to just share a little bit about that. So welcome to our webinar. Next thing we wanted to share with you guys is that the webinars are part of the CARE program. So the LGMD CARE program is a new initiative with the Speak Foundation with the purpose to help identify newly diagnosed patients with limb girdle muscular dystrophy. One of the things that we have found is that newly diagnosed patients don't have an easy on-ramp to find the existing limb girdle community. They may get diagnosed and flounder around for a while, and they don't know how to necessarily plug in. So this CARE program is designed to identify the newly diagnosed and to connect and reach everyone. Another purpose of the CARE program is to educate through these webinars and also to inform patients about new and upcoming clinical trials. To us, this CARE program is very important. I wanted to also share we also have some additional grants available for our COVID-19 relief program through the Speak Foundation. If you are in need of a grant, the grants are now up to $400 for grocery assistance, and you can actually apply for that on our website, and that is at speakfoundation.com. You can also engage with us at nationallgmdconference.com as well, and that is the National Limb Girdle Conference. And GRASP LGMD has plans for being a part of that next year as well. And this past year in 2019, we had a great conference, and our hopes and plans are to do that in 2021. We are communicating about having a virtual event in the event that COVID-19 is still a big issue. So we will let you know more about that and update you more as things get closer. Next, I want to tell you that this webinar is sponsored by Sarepta Therapeutics. We are so grateful and thankful for Sarepta Therapeutics and their support of the Limb Girdle Muscular Dystrophy community. Sarepta Therapeutics has some great ways for you to stay connected. In case you didn't know, Sarepta is a global biotechnology company on an urgent mission, engineering precision genetic medicine to reclaim futures otherwise impacted or cut short by rare disease. You can stay connected with Sarepta through an education and mailing list. If you're an LGMD patient or caregiver, a resident of the U.S., and would like to receive updates from Sarepta Therapeutics, please visit limbgirdle.com and sign up to allow future communication. Or you can email. If you wish to direct or connect to someone from the Sarepta Patient Affairs team, you are welcome to email advocacy@sarepta.com. And we are so grateful and thankful to our sponsor. At this time, I am very pleased and so honored that Dr. Nick Johnson is on with us. Dr. Nick Johnson is the GRASP LGMD founder. He is also an associate professor of neurology and human and molecular genetics and vice chair of research in neurology at Virginia Commonwealth University, with a focus in inherited neuromuscular disorders. At this time, I am going to welcome Dr. Johnson to begin sharing his slide deck, and after he does his presentation, we will come on with a short Q&A from patients who pre-submitted questions. Thank you so much, Dr. Johnson, for being with us, and I am excited to have you on.
Thanks so much for having me today. And the first thing I'd like to say is I'm hoping that all of the members of the community who are out are staying safe at home and being, well during the course of this public health emergency. And I'll talk a little bit more about that later. So I thought it was timely to talk about where we are. I mean, I think we all left the last National LGMD Patient Conference with a lot of hope and inspiration, thinking about the development of clinical trials and programs over the course of the next 12 to 24 months. And I know there's a lot of questions out there about where we stand, again, in the face of this pandemic. So I will, first, these are my disclosures. I'm going to start by reminding us all where we started, or where we were pre-COVID-19 in terms of clinical trials. And the list that I have here includes not a comprehensive list, but these are trials that have been registered in some way or another on clinicaltrials.gov. And I would just highly recommend that that's a great way for you to find what studies you may be eligible for. So you can see there are a number of studies conducted by our consortium, the GRASP LGMD Consortium, including a focus on LGMD 2A, 2B, 2C through F, 2I, 2L, and 1D. Nationwide Children's has their own natural history studies. There is, of course, the University of Iowa's Wellstone Center, which is a long-standing natural history study in alpha-dystroglycanopathies, NFKRP. There are natural history studies of LGMD2I in Denmark, France, and the United Kingdom. There's the Global FKRP Registry, and then there's the COS2 study, which focuses on LGMD2B. And there's a single treatment study that's listed on clinicaltrials.gov, which is the gene delivery trial for LGMD2E. And so, it's certainly striking when I looked at that list. There's a long list of natural history studies and a short list of treatment trials. And I think everybody on this call would really prefer that list was shifted in terms of longer treatment trials and fewer natural history studies. But I think before we even think about that, it's worth thinking how you can help drive that balance forward, drive us towards successful precision-based therapies in limb girdle muscular dystrophy. And so just as a reminder to all of us that are on the call and looking at this, how do you participate in research? How are you supposed to get involved? We want you to be involved. I think many of you want to be involved and help move these treatments forward. So what are some things that you can do? So one thing that I highly recommend is looking for studies on clinicaltrials.gov. This is a federal database that is a repository for clinical trial opportunities that includes natural history studies and treatment trials. It does often include information about what age you need to be. So can you be a child or an adult, or what ages are required for eligibility, what subtypes of limb girdle muscular dystrophy are included. And then sometimes there's information about the functional status. So can you use a wheelchair? Do you need to be walking still? Sometimes it may be difficult for you looking at that to just think about whether or not you would qualify or not qualify. But I'll jump to my last bullet point there, which is that if you're not sure if you qualify for the study, or if you're not sure about the risks, or if you need more details, just ask. Just ask. That's what. There's lots of people that you can ask if you qualify for that study, or if you have additional questions about whether or not that's the right study that's fit for you. Those studies include contact information. It's often either the principal investigator, who's the person supervising the study, or a clinical research coordinator, who's often the person doing a lot of the study conduct and logistics. And there's email or phone numbers. Pick up the phone or write an email to that person and say, hey, I'd like a little bit more information. And I get those emails and calls all the time, and I'm certainly happy to look through and say, yeah, we think you'd be a great fit, or maybe this isn't the right study for you. So that's participating in Research 101. That's a good place to start. But, continue on that thought, what study should I participate in? I think that this is the beginning of this question for our disease community. This is the beginning of that question for limb girdle muscular dystrophy. Because right now the question is, which study should I participate in? And there may be more than one natural history study. What I'm hopeful for, we get to a place where we're able to say, well, which treatment trial should I participate in? That's an exciting conversation to have. And so but it's worth laying the foundations now and how we think about that. So the first thing I'll say is that for the natural history study, many people can participate in more than one natural history study. So just because you do one study doesn't mean you can't do more than one. They're not mutually exclusive. I'll talk about what some of the differences are in a slide or two. But certainly, there's no reason why you couldn't do more than one. And I would encourage you to do more than one if you were able to do that. I'm hopeful that your neuromuscular expert can guide you to the best studies for you specifically. Often, I'll tell you right now, the neuromuscular physician community is one. I know many experts across the country, and so they can certainly guide you about which studies might be most appropriate for you. And then, we're not there yet, but while we're having this conversation, it's worth remembering that drug trials include more risk than a natural history study. And so you certainly need to discuss with the investigator what those are and if they're acceptable to you. So oftentimes, particularly if you're traveling to a site coming from a distance, I know our sites and many other investigators are happy to provide you kind of a basic list of the risks and benefits to you over the phone or by email, so that you're able to consider that and thoughtfully think about whether or not that's something that you want to participate in before you get in a plane or drive 6 hours to a particular site. So that's at least how I think about what study you should participate in. I would hope that you participate in a study. We want you to help because it's certainly a partnership. So why do a natural history study anyways? Or why are there so many natural history studies? I know I've been asked that question as of late. And I think part of the challenge is on us. It's our terminology. Natural history studies is kind of it's a big bucket. It talks about a lot of different types of studies. So some of these studies look at different patient groups. Some of these studies are designed to look at Alka measures, or how we measure your function. Some of these studies are not really designed to look at any of that. They're designed to look at how, a biomarker such as muscle MRI, or taking a piece of muscle and looking at it under a microscope, how that performs. So there are a lot of different kinds of natural history studies. And when you create a natural history study, you're doing that with a specific objective. And so it's important to remember that for a particular subtype of limb girdle muscular dystrophy, one natural history study may be really geared towards design, thinking about muscle MRI. And another natural history study may be designed to look at people who are very young with the disease. And another one may be geared at looking at, in somebody who's, still walking, how fast does your walking change over the course of a year? So you can see that there's just lots of different questions that you can ask from the same type of study. And one of the real challenges is, depending on what question you ask, depends on how you design that study. How many people are included? What age groups are included? Do people have to be walking to be included, or can they be non-ambulatory? So I think that's probably the main driving force as to why there are so many different types of natural history studies. It's hard to bucket and package that all into one category. And then the last thing I'll say is that I wish that we were so intelligent the first time we do a natural history study that we answered all of the questions that we needed to the first time. But I have to admit that it's frequently the case that we look at the data over the course of 12 months, and we say, actually, you know what, we're missing this part, or what about this, or we really should have been looking at this particular part of it. So, and unfortunately, sometimes there's a bit of a process to get to the right place. And I think that the right way is to develop those iterations and to think about the study over the course of a natural history study, because when you're doing a drug treatment trial, there is only one drug that you're trying to see whether or not it's effective. And so I'd much rather make sure that we get it right, that we design very effective, efficient clinical trials, so that when we have those really exciting new therapies, we're not saying, well, we don't think the study design was appropriate to be able to see whether or not it worked. So I think that's the reason why we're taking our time. Maybe that's not the right word. We're not taking our time, but we're spending time and making sure that these natural history studies and these tools that we develop for clinical trials are appropriate. So those, as I said, those natural history studies do provide tools for clinical trials. I'm cognizant of the fact that sometimes we use terms, and I want to make sure that we're talking about the same thing. So Alka measures are tools that we use to tell if something in limb girdle has changed between 2 points in time. They can be lots of different things. Sometimes they are something that you can measure in the blood or muscle, in which case we call them biomarkers. Or they could be something that a physical therapist or clinical evaluator assesses, like strength or the ability to do tasks. And these are often called functional outcome measures. Or they can be questionnaires that describe how you experience what your experience is of limb girdle muscular dystrophy. And we spend a lot of time thinking about those outcome measures and what are the characteristics we're looking for. We're looking for things that are pertinent to limb girdle, that they're reliable, that they measure same thing one day and you measure it the next. We want them to be sensitive to progression. We recognize that this is often a very slowly progressive condition, and that we don't want to spend 3 years trying to see whether or not a drug works. We want to see, if we can detect that change in the shortest period possible, that they're inexpensive, and that they're feasible to collect at multiple sites. And the last point is that we have not settled on the right mix of outcome measures for limb girdle muscular dystrophy yet. I'm hopeful that a number of the studies that I showed on that first or second slide, looking at the different natural history studies, that we'll be able to come to some conclusions, definitive conclusions at the end of those studies, and move on to treatment trials when they're available. So you do need the right tool for the right job. They are just tools that we use to get drugs approved in people, but we need the right tool. For example, measuring gene expression won't tell you whether or not somebody's experiencing pain. Those different tools are actually important at different times in the drug development process. And so we think a lot about developing what we call a clinical trial toolbox. So you can't always use a hammer to get a screw into the wall. So you need a hammer, a screwdriver, tape measure, et cetera. So we want to make sure that we build all the right tools to build that house for you. Just to briefly say, and you'll hear from, I'm really super excited actually about this partnership with Speak Foundation and our ability for a lot of the investigators in our consortium to share the knowledge that they have with the broader Limb Girdle Muscular Dystrophy Consortium. So this is the GRASP LGMD study, so genetic resolution and assessment solving phenotypes. It's a lot easier to say GRASP than the acronym, or than the words that the acronyms are composed from. So the concept behind this is that this is actually a platform natural history study. So we are intent on using the same outcome measures at the same sites with the same training speeds, training mechanisms to be able to study new subtypes. And so our goal is that the outcome measures that we develop for 2A can be transferred to 2I or vice versa, or 2L, and so that we're not having to spend time with each new subtype and really go back to the beginning of the drawing board. And really the goal is to speed the development of and validation of these outcome measures for upcoming clinical trials. All of the studies that we have ongoing are 12 months in duration as it stands right now. Many of the visits include a survey, measurements of your muscle strength and function. They all include a blood draw, and depending on which particular subtype you have, your study visit may include a muscle biopsy at a couple of the visits, or a muscle MRI to look at the lower extremity muscles. So those two depend a bit on what subtype you have and whether or not you're walking or not. So you can see there a list of the subtypes that we are currently enrolling across the different natural history studies, the gene mutations associated, and of course, I would be remiss if I did not include our sponsors who are driving these studies. So a big shout out to the Muscular Dystrophy Association and C3 Coalition of Cure Calpainopathies for providing the initial funding to get many of these studies off the ground, National Institutes of Health, and then the MDA, Sarepta, and we're sponsoring specific subtype studies. So with that, I think that's what I would say about our studies. We have tried as best as possible across our consortium to be geographically dispersed. And so we have sites across, I'm not going to read all these sites off to you, but there's a fair number of sites up and down the Eastern Seaboard and Midwest. I apologize in advance that once you kind of head west, there are sites in Denver and Irvine, so it's a little bit sparse, and then a few sites for studies in Europe at either Newcastle or Copenhagen. I want to specifically call out Lindsay Alfano as our lead clinical evaluator, who's done a lot of work to help develop and validate those functional outcome measures along with Linda Lowe's. And then, of course, Chris Weil at WashU, who's spearheading our biobank and repository, and then Jeff Statlin, who's been helping with the overall trial design. So still pre-COVID, I talked a lot about why we're doing natural history studies, and it's certainly interesting and good to do that, but it's, the real excitement is to prepare these for therapeutic trial development. And I'll touch on a couple different areas of therapeutic development that are really close to being ready for in-person or in-human clinical trials. They're still largely what I would call preclinical, so you're finishing up a lot of the safety testing, and that includes gene replacement therapy and what I call small molecule approaches. So gene replacement therapy, the best way of thinking about this is that these trials, or the design behind this, is to take a virus that's typically not naturally occurring. So they often use some form of an adenovirus or synthetic adenovirus. You take out all the bad viral DNA or RNA, and you stick in the gene that's missing. And that virus gets injected and then allows the defective or the new gene to come in to the cells and muscles, and it replaces the defective gene. This type of therapy is best in autosomal recessive conditions. For those of you who know that that's autosomal recessive, or who have a limb girdle muscular dystrophy that's a 2, or using the new nomenclature LGMD-R or LGMD2, these are really the optimal places to be thinking about gene replacement therapy, because you're missing a gene, and then the idea here is that it's restoring it. There are technologies designed to do with gene toxicity, which is often what happens in autosomal dominant limb girdle muscular dystrophies, but I'm not going to touch on it during this particular talk. These gene replacement therapies, as we understand it, are currently likely one-time therapies. They do carry a potential risk of immune reaction because you're injecting a virus that you've not been exposed to. It's like a very, very large vaccine. And so that carries that. There's lots of steps and techniques that investigators have developed to mitigate that risk. But just to at least call out kind of the pluses and minuses of that particular type of therapy approach. And I'll show you some data that's been published out of the Columbus group, the Center for Gene Therapy there at Nationwide Children's. And this is all looking in mice at what happens, and I'll try and show it to you pictographically here. So this is a mouse here with LGMD2E, so to speak. And then after they've provided the AAV sarcoglycan beta gene therapy, you can see, as soon as I get my mouse to work here, you can see that the proteins are now staining on the outside of the muscle. And so it's really impressive that you can see that those fibers have come back and showed there. So a good response in those mice. So you can see that the gene or the muscle protein comes back, and that's important. But equally important, what happens. I particularly like specific force measure over here, and which is a measurement of mouse strength. And you can see that these sarcoglycan beta knockout mice have reduced force, and that as you add the gene therapy vector, it comes back. There are a number of gene replacement therapy programs in development. These are the ones that are publicly announced. You can see these either at sarepta.com or askbio.com. So the clinical programs are those that are in early phase clinical trial testing. So these are often safety measurements in sarcoglycan beta, alpha, and dysferlin. And then there's a number of preclinical. So these are ones where the gene therapy product is either being tested and optimizing the exact components, or it's being tested for safety, often in animal models, in sarcoglycan gamma, ANO5, calpain 3, or FKRP. So small molecule approaches are a little bit different. So this is what most of you are thinking about when you're thinking about drug development. This is like aspirin or Tylenol or really nearly any other medicine you've ever taken. These are often easy to deliver. They're often oral, so you not by an IV or other injection, and don't typically act specifically on the missing protein, but often act on the downstream actions from the loss of that protein. So one good example of this is in 2I. You can see with 2I, you have this loss of an enzyme called FKRP, and that prevents the incomplete glycosylation of a protein called alpha dystroglycan. But it turns out, actually, if you can provide this exogenously through a synthetically derived component called ribitol, you can actually drive that glycosylation pathway and improve in mouse models the glycosylation of the alpha dystroglycan. So again, this is another example similar to the gene therapy slide, where you can see that the treatment of the mouse improves strength. So I'll look down here. So you've got fibrosis, which is a measure of scarring in the mouse, and you can see that it's reduced with the compound that's delivered both in the muscle and in the diaphragm muscle, and that the strength and function is actually improved as well. So we made a lot of progress in terms of developing new therapies that are being tested in animals for safety and getting ready for almost to the point of human clinical trials, and combating that with the development of natural history studies to develop those outcome measures. And then we had COVID-19. And so before I step into what we've been trying, how the community, the research academic community has pivoted, I just like to remind everybody that COVID-19 is caused by a SARS coronavirus. It leads to fever, cough, and respiratory distress. That people with underlying medical conditions are at higher risk of complications. And that includes people who have muscle weakness of the chest wall or the diaphragm, people who have a weak cough or cardiac involvement. And so, unfortunately, that's a lot of people with limb girdle muscular dystrophy. And so our advice is to follow the guidelines that have been issued in your region for sensitive individuals or people at risk. So the Muscular Dystrophy Association has guidelines that they publish on their website. I know the Speak Foundation has offered some guidelines and resources there. The World Muscle and the CDC all have guidelines that talk about what you would do if you're in an at-risk group. So first thing I'll say is stay safe. So on our side, because of the public health emergency, nearly all universities have halted in-person research visits across the United States and Europe through most of the spring and early summer. And unfortunately, that's necessarily a delay in in-person studies for limb girdle muscular. And on top of that, additional travel bans or new challenges. For example, all of our investigator meetings, as it stands right now, have been conducted by Zoom, which if you've ever tried to sit in front of Zoom for about 8 hours, you realize you cannot do that. And so it's made things a little bit less efficient in terms of being able to meet in person, which is typically how we can really move the progress forward. And so the group of GRASP LGMD investigators have been thinking, what are we going to do? How are we going to move this forward? We don't want to waste time and delay these studies. And so lots of credit again to Lindsay Alfano and Jeff Statlin and Brittany Holmberg, who's our project manager, for coming together and thinking about ways that we can solve this. So can we do this virtually? Can we do your study visits, not where we see each other face to face, but over Zoom or other video conferencing software? And we're working on that. We think that's really important. So the first thing that we need to do is that we need to make sure that the data that we collect, so how we measure your strength and function by video camera, looks the same and it measures the same as how we would measure it if we were in person. And so that's the big hurdle that our group is thinking about right now, which is reliability of those measurements. And so we do have plans upcoming in the very near future to transition these studies to what we call a virtual pilot, which is those visits will not necessarily begin face-to-face in the clinic, but rather will begin in your living room or kitchen and my upstairs bedroom. And you'll be able to actually enroll in the studies virtually. We can still perform the surveys that we do as part of our work. We think we can still measure function. Unfortunately, we can't collect some of those important biomarkers. So even with all of the work that we're doing now, we think that there will be a time and a place where we'll need to get you to come back into the clinic. But while it's still, I think it's not clear, many states still have travel bans and stay-at-home restrictions depending on where you are today. I know that's been changing week to week. During that time, we're focusing on being able to keep you safe at home, and hopefully over the summer and fall, we'll be able to get you back into clinics. So if you are interested in enrolling in those consortium studies, you can contact Brittany Holmberg, our project manager. My email is here. Feel free to contact me directly. We will direct you to the closest or preferred sites. So I'll often provide you, or Brittany will, a list of particular options, because sometimes it's easier to drive north or south or east or west, so to speak, even if a site's a little bit further away. We believe that clinical trials are likely to remain on current timelines. I think a lot of the regulatory and safety work has been ongoing, even during this public health emergency. We will certainly continue to work with the Speak Foundation and our pharma partners to announce clinical trial opportunities once these therapeutic trials begin, and as they're appropriate to announce, with the Speak Foundation. So we remain hopeful that we haven't lost a lot of ground in terms of the timing and approach for those treatment trials. One last thing I'll say is that while you're waiting to do other stuff, including enroll in these some of these virtual pilots, there is a survey project that's been ongoing with Dr. Statlin and Dr. Twill at the University of Rochester, that seeks to understand how you're getting medical care right now, as well as the psychological stress of the public health emergency. There's a link to the REDCap survey here. I believe you should have received it, I think, last week or earlier this week from an email from the Speak Foundation as well. So we very much appreciate you completing the survey while you're waiting to do some of the other stuff in your spare time. So with that, I want to thank all of our investigators across the United States and Europe. I want to thank our sponsors, National Institutes of Health, MDA, Coalition Cure Calpainopathy, ML Biosolutions, and Sarepta, and I will take questions.
Thank you, Dr. Johnson. That is so wonderful to hear all of this information that you've put together.
Perfect.
All right, so I've got some questions, and we try and obviously, as we've had our presentation so far, I'm getting a lot of people sending me like emails and texts. One of the things that you had already mentioned, but people can join remotely to start some of these clinical trials. Is that the case? That's what I'm gathering from what you said?
So it's not quite ready yet, but the goal of the GRASP LGMD consortium moving forward into June would be that we would allow for remote enrollment. That's correct. So if you contact us now, we'll put your name on a list. And then as that is ready to go, we'll contact you and you can just enroll and start remotely. That's correct.
Okay, that's wonderful. How do I know, this was a pre-submitted question, how do I know if I'm going to be excluded from a trial because I don't meet the qualifications required to be in it?
Yeah, that's a tough question. That's always tough to hear too. I have to say, it's tough to say we can't take you into the trial. If it was up to me, we'd take everybody. But sometimes that's just not possible. And so there's a couple ways you can get it. Some of it, like I said, if you look at the clinicaltrials.gov site, that will often have some of the basic information about what would make you qualified or disqualified for a particular study. So that's a good place to get that information. And then, like I said, if you reach out to the study contact, they can often take some of your information and screen to see whether or not you'd be qualified for that particular study. And they're all in the end. There's no same answer. All the trials have different inclusion exclusion criteria.
Okay. Okay. Thank you. Do you think, do you have any information on when a trial for 2D R3 is expected? And is there a particular group working on it?
Yeah, that's a great question. We are expecting in our consortium to start a natural history study in the very near future. So please reach out to us if that's your subtype. We'd be happy to enroll you in that study. I can't say in terms of when a treatment trial would be ready, but I do know, and it's available on the Sarepta website, you can see that they're working on that particular subtype in terms of developing that gene replacement therapy.
And I just wanted to add to the nomenclature for limb girdle is changing. So I think for everyone who's new to limb girdle, if they're newly diagnosed, you might see a different reference to your particular subtype. And I just wanted to share that if you see the different designations, not to get confused, but the 2D R3 is indicative of that new nomenclature, correct?
Yeah, so the 2D is the old and the R3 is the new. So I've been saying a lot of it the same. And to be honest, if you're not sure, just say the gene name.
Yeah, there you go. That's a good way to say it. In addition to existing trials, one of our patients submitted this question. I'm interested in whether Dr. Johnson's research unit or even others is preparing research on the mental health status of those with muscular dystrophy during the pandemic. So I do think that this is in reference to perhaps the study that we're seeing at University of Rochester. Is that correct?
Yeah, so there that's a great opportunity. There's a questionnaire about perceived stress, and there's data that's been collected prior to the pandemic about what normal perceived stress might be. So that's a good way, and something that we've been doing in collaboration with the University of Rochester to see what the mental health effects of the pandemic are.
And I was going to add on that too, we will actually load up all the information on this particular survey onto the Speak Foundation website. So if patients wanted to get to that link that you had shared a little bit earlier, that'll be on our website. So they could go there just so they could get it easily and click on it if they want to enroll in that survey. And then we had another question that was submitted. Can a mother have one type of limb girdle muscular dystrophy and her children have another type?
So that would be unusual. Typically speaking, the type of limb girdle muscular dystrophy that would be kind of running in families would often be the same type. It's not impossible. For example, if the partner had a different type of muscular dystrophy, then the genetics become complicated. So for that particular question, sometimes it's best to speak with your care provider or genetic counselor to kind of understand what that situation is. But commonly speaking, if that's your type of limb girdle muscular dystrophy, that might be what you might pass on to your children. And the last thing I'll say about that is that that also depends on whether or not it's autosomal dominant or recessive, and what your relative risk is of passing it on to your children. But I would definitely take that up with your care provider.
Okay. Is it okay to say something very simple in terms of if you have a recessive form, the only way for you to pass the actual disease on to your children is if you marry someone and have children with someone that's also a carrier? That's
perfect. That's correct. Yes.
Okay. I just wanted to make sure I was correct in saying that.
Yeah.
Saying that for a few years. Okay.
Right.
All right. And then and this was a question submitted, and I've heard this asked a couple of times, not very often, but someone asked, have you ever seen any cognitive issues in a person with limb girdle muscular dystrophy?
Yeah, that's a good question. So it's not, it's by far and away not a defining feature of limb girdle muscular dystrophy to have cognitive issues associated with it. In the old nomenclature, there were some types of autosomal dominant limb girdle muscular dystrophy that had some cognitive features that could be associated with it. Those were quite rare to begin with. And in the new nomenclature, they're not considered limb girdle muscular dystrophies anymore. So, so the short answer is probably 99% of the time, the answer is no.
Okay. And I know in a future webinar, we're going to discuss VUS, the issue of VUS. And so I'm excited about that one in the future. So that to me is just a very interesting webinar as well. And I'm excited that we'll be able to talk about that. And I know you have particular interest in that area as well.
Yep. Yeah, that's the VUS is actually it's a big deal. It's something we need to solve as we get into the treatment trial era, because it's unlikely that a VUS would qualify for a trial. And so I know members of our consortium are certainly working on reducing the number of VUS that are identified, or getting them to either be classified as benign or disease causing. So it's an important issue.
You know, it really is. And I know a lot of patients will submit questions and ask about VUS. And so I know it's a concern for many patients. And so I'm excited that we'll be able to have that as a future webinar. And I think at this point, I think we're at the end of our questions. Now, I wanted to share with our audience, if you have a question that wasn't answered today, Dr. Johnson has also offered to answer some of those questions. So if you'd like to submit some additional questions, you can send them to Catherine at thespeakfoundation.com, and we'll be glad to send those questions and send them over to Dr. Johnson, and hopefully we could get some responses to you in a good length of time. I know I've received lots of emails with lots of questions, and we are so thankful for anybody that's on this call, and we know how important it is to get these answers. And so I myself have limb girdle muscular dystrophy type 2I. So I myself have limb girdle muscular dystrophy. So I know what it's like to deal with this disease. And so I just wanted to tell you so much. If you're on this video conference and you're dealing with limb girdle, I know what that feels like, and we're together. So thank you so much, Dr. Johnson, for being on this call with us, and we appreciate all of the slides and the information you presented.
Happy to help. And like I said, stay safe.
Thank you so much. And I want to say thank you again to Sarepta Therapeutics for sponsoring this webinar. And this is the first in our series in conjunction with Limb Girdle Muscular Dystrophy and the GRASP Consortium. Thank you so much, Dr. Johnson.